We are now moving into our third portion of this large chapter called cellular reproduction. But before we move into our last chapter, which is about meiosis, I'd like to compare the difference between what we've done and what is to come. So obviously we have already discussed the term mitosis, which will eventually result in two identical offspring, which are identical to the original parent. And now we are moving into a new process called meiosis, which will result in four different haploid cells. So let's get started. What is meiosis? It is the division of the nucleus, which is exactly the same thing as mitosis. It was still the division of the nucleus, but now the difference is that it's going to reduce the number of chromosomes by half. So if you have an original cell, which would be a diploid cell, and you reduce that number by half, you've now created a haploid cell. This only happens in sexual reproduction. Mitosis, which is what we've discussed before, only happens in asexual reproduction, so there's another difference. Mitosis produces body cells, and meiosis produces the sex cells or the gametes. So the only product of meiosis is either going to be a sperm cell or an egg cell. So let's take an overview of meiosis. Let's see what is similar to mitosis and what's different. So as you can see here, we're starting off with our diploid cell. Remember, 2N stands for diploid, 46 stands for how many individual chromosomes are found in the cell. So you have an original diploid cell. Now, the difference from mitosis to meiosis is that during the S phase of this cell, the 46 was temporarily duplicated to 92 in order for it to be split to get identical 46 and 46. And that's how the identical cell was created. In meiosis, the DNA does not duplicate in the S phase. That's the major part of this. So when you split 46 in half, there is no duplication, so it stays at 46. Half of 46 is 23, and the other half is 23. Notice how the 2N, which means diploid, reduces to N for both of these, which now means haploid. Now we call this process meiosis I, or PMAT1. PMAT standing for prophase, metaphase, anaphase, and telophase. These phases still exist, except for when you're talking about meiosis I, you need to put a Roman numeral 1 after every single phase. So there's prophase 1, metaphase 1, anaphase 1, and telophase 1. If the 1 is missing, then you're referring to mitosis, which would be incorrect if you were talking about meiosis. So this is the first division, and the goal of the meiosis I division is to turn the diploid cell, is to reduce the amount of chromosomes and to turn them into two haploid cells. That is the goal, the end result of meiosis I. Now we have another division. Mitosis doesn't have two divisions. Mitosis has one division, which we said results in identical cell, and then the cycle continues on and on and on. Very different in meiosis. We have two divisions in order to get the product that we want. So now, this portion right here is very similar to mitosis in terms of we have chromosomes lining up, we have chromatids that are splitting, and your result is an identical cell, or two identical cells, to the original parent cell. The only difference from meiosis II and mitosis is that the original parent cell has 23 chromosomes, whereas in mitosis, the original parent cell had 46. So the goal of meiosis II is not to reduce the chromosomes, because we've already done that in meiosis I. The result in meiosis II is just to form more of them. So instead of having 2 haploid cells, we now have 4. Now, because this is meiosis II and the cell still goes through prophase, metaphase, anaphase, and telophase, in order to distinguish between the difference, we now have to put a Roman numeral 2 after each phase, so it would be considered prophase 2, metaphase 2, anaphase 2, and telophase 2. Again, if the Roman numeral is dropped, then you are talking now about mitosis, and therefore that would be incorrect. So here is a more detailed picture of what's happening in meiosis I. Remember, the goal of meiosis I is to take an original diploid cell and to reduce the amount of chromosomes and make 2 haploid cells. So what we see here, and this is the major difference that you'll notice between mitosis and meiosis, is that in meiosis I, instead of having chromosomes line up, you now have homologous chromosomes lining up. So they're not going to be found in a nice, neat row of single row, meaning like 1 chromosome here, 1 here, 1 here, 1 here. Now they are going to be paired, and they will always be paired. And remember, homologous chromosomes are 2 chromosomes that carry the same genes, they have similar shape, and they have similar size. So they're going to find each other. They're going to line up here along the metaphase line. So when you describe that, you wouldn't say chromosomes line up, you would say homologous chromosomes line up or homologous pairs line up. Then during anaphase, you wouldn't say chromatids split, right, like we did in mitosis, but now you would say chromosomes split, because you have a full chromosome going this way. You have a full chromosome going this way, and then the pair, the full chromosome, is going to the other end. And now what you've done is you have reduced in half the amount of chromosomes for one cell to the other, this one now having 23, this one now having 23, when the original parent cell had 46. Now entering meiosis II, the 2 original cells that you have going in are 2 haploid cells, both containing 23. Now we can say the same exact terms that we did for mitosis, where we say chromosomes line up, chromatids separate. Remember, chromatid is half of a chromosome. And now we have our haploid, 4 haploid cells. So during meiosis I, prophase I has a lot of different things that go on. The first major thing is that we have homologous chromosomes lining up, or starting to line up, right, getting to the point of metaphase, but they're trying to pair themselves up with their pair, and that's their homologous pair. Remember, they're the ones that have same size, same shape, and they carry the same genes. Now, when they pair up, they form something called a synapsis, which is just the 2 homologous pairs finding each other. It's just a point at which they find each other. And when they do that, they form something called a tetrad, which is just, well, I'll show you in the next picture. It's going to be a little bit easier for me to explain that on the next page. But all of this right here, just understand that the homologous chromosomes need to find each other before they start lining up in metaphase. That's the major thing I'd like you to get out of that. Same as mitosis, the spindle fibers are going to appear coming out of the centrioles, and crossing over, this is a new term that we haven't talked about, crossing over may occur. It is a completely random process, and I'm going to talk a little bit more about crossing over in the next slide or two. So first, let me just show you the synapsis and the tetrad like I showed you before. So here are this is one homologous pair. Here is one full chromosome. Here is another full chromosome. Together they form a homologous pair. And when they meet, they form a synapsis. So that's the point at which their chromatids overlap. And when that point happens, they now form this portion right here, which would be 1, 2, 3. That's a tetrad, and the term tetrad associates with the number 3. Now if we take a look here, this is what crossing over is. Crossing over is an exchange of the genetic material between the homologous chromosomes. This is not a guarantee if it happens at all. This may happen once, this could happen 10 times, and it can happen, it can cross over and then cross back, it can cross over in multiple areas and cross at the top, across the bottom, but it may never cross at all. It's completely random. It really just depends on how close the homologous pairs get to each other. It depends if they form a tetrad. It depends if they meet at their synapsis. But when this happens, you have an exchange of genetic material. So let's just say, for example, the red chromosome is coming from dad, and the green chromosome is coming from mom. So if you have an exchange like we see here, so here would be the point of synapsis, right? This is our tetrad. And now this piece of the green chromosome has now attached itself to the red, and the red has now attached itself to the green. Now this is good, this is okay, nothing terrible happens. All this does is this just gives a little bit of mom's genes on dad's chromosome and a little bit of dad's genes on mom's chromosome. But remember, because they're homologous chromosomes, they carry the same genes in the same spot. So, you know, maybe this is the gene for eye color and mom has blue eyes and dad has green eyes, and you've just switched the color of the eye for whatever chromosome would get chosen at the end. Here's a small animation of crossing over happening in action. So if we start at the beginning, here we have them lining up, crossing over, and then they pull apart. Okay? Actually, this whole thing is meiosis. So, and then it goes really fast, so it's harder for me to commentate it as it's going, but there's crossing over. That's the end of meiosis I, and this is meiosis II, and you end up with 1, 2, 3, 4 haploid cells at the end. Notice how by the end of meiosis II, you have lots of mixed up colors. You could get this cell or this cell or this cell or this cell, and all of them are prime candidates for joining up with the other sex. So if these were sperm, any 4 of them can be chosen, only one of them can be, and another one could be an egg, and depending on which genetic variation of genes that they get will depend on what that new organism, what characteristics they're going to have. So this is what increases genetic variation. This is why you and your brothers and your sisters and your mom and your dad, this is why we are not clones of each other. This is why we all look different. That's why there's no one on this earth that looks exactly like you. Even if you're an identical twin, there's still going to be some type of difference or variation. And this is why. It's because of crossing over, and it's because of the exchange of the genetic information and the choice of which one will get chosen. All random. So as I stated before in the past couple slides, I said that crossing over would increase genetic variation, and we just simply call that term genetic recombination. And all that really means is the shuffling of genes and the random choosing of whatever cell gets chosen which has the shuffled genes just increases genetic variability, and with that, that allows for the offspring to look different and to have different traits than the parents, which is a positive advantage for survival, because if a gene is successful, that gives that individual more of an edge or more of a percentage of being able to live to the point of being able to have their own offspring, and therefore those positive genes are going to be passed down. Okay, moving on to metaphase 1. So everything we've been talking about so far has been all about prophase 1. Prophase 1 is super important in terms of crossing over, in terms of the homologous chromosomes finding each other, forming tetrads and synapsis. But now, once all of that has occurred, now they just simply have to line up. And remember, they're not lining up as chromosomes, they're lining up as homologous pairs. So this right here is a homologous pair. This right here is a homologous pair. Notice crossing over has already occurred because we have a little bit of the blue on the red and the red on the blue, and vice versa. So the same concept of metaphase lining up in the middle. Difference now, it's just a different term of saying that homologous chromosomes have lined up versus just chromosomes. Okay, same thing with anaphase 1. The only difference, it's the same concept where they're being pulled apart from, from the middle, being pulled apart to one side, being pulled apart to the other. The difference is in the vocab. Instead of saying chromatids are being pulled apart, now we are seeing full chromosomes are being pulled apart. So one chromosome goes one way, one chromosome goes another. Remember, this right here is the halving or the reducing in half of the amount of chromosomes from 46 to 23. This is how it happens. You're literally splitting them in half. And telophase 1 is, again, very, very similar process to mitosis, where we see the beginning of cytokinesis. We have the nuclear membrane coming back, the spindle fibers are receding. You now have the chromosomes turning back to chromatin, which is the spaghetti, and it's ready to undergo meiosis II. As stated before, meiosis II is very similar to mitosis. The only difference is that the parent cell in mitosis has 46 chromosomes, and you want to end up with 46 as an identical. And in meiosis II, the parent cell has 23 chromosomes, and you want to end up with the identical of 23. But other than that, the stages are the same, except for you throw a Roman numeral 2 after them. You use the same exact vocab because the same things are happening. So in terms of vocab, it's very similar, except for the number of chromosomes. So here's the product of meiosis, completely the end of meiosis II. You want to have 1, 2, 3, 4 genetically different haploid cells, genetically different from the original parent cell. And remember, this results in only 2 types of cells. These are the only sex cells that exist: human sperm cells and human egg cells. Now, the difference between the production of sperm and the production of eggs is that during the process of spermatogenesis, which is just the making of sperm, if you are a male, you go through the process of meiosis and you produce 4 sperm cells, which is exactly like we said, going through meiosis I, having the diploid cell, going through meiosis II, and gaining 4 haploid cells. Okay, pretty simple. But for females, it's a little bit more complicated, and a process called oogenesis. So an ovum is the term for an egg cell, and when we look at the process of oogenesis, instead of producing 4 cells, which we do, we go through the process and we have the 4 haploid cells. Unfortunately, only one of them is functional. So if we take a look at this picture here, okay, comparing spermatogenesis, which is the production of sperm, and oogenesis, which is the production of the egg, this is very cut and dry, right? You have your diploid cell, you've halved it, now you have 2 haploids, and now you've gone through meiosis II, and now you have 4 haploids, which then develop to become 4 sperm cells. Females have meiosis I occur, except for only one of the eggs is functional. They both go through meiosis II, but again, only one of them continues to be functional. These just die off, they're not used. And then we have the differentiation of the egg cell into the fully functional egg. So females go through the whole process. They do form 4, but only one is functional, so 1 functioning egg, where males produce 4 sperm cells per round of meiosis. Once the sperm and the egg are produced, they have to fertilize. So the sperm, whichever sperm will make it go through the actual cell membrane of the egg, and it will reach the nucleus. If you see here, we have the nucleus of the egg and the nucleus of the sperm. I don't know exactly which one is which because they look very similar, but once they join together and they fuse, that is when this whole cell becomes diploid, because the sperm has now brought 23 chromosomes. The egg had 23 chromosomes. 23 plus 23 is 46. And now what happens in here is a bunch of mitosis, right? Because now we want to keep the 46. We don't want to divide it again. So we want to keep dividing, and growing this 46. And that is when you have your fetus, and eventually it becomes baby. And here is just an overview of possibilities of the arrangement of chromosomes, the homologous pairs. So we call this the law of independent assortment, and that just simply means that it's completely random how the homologous pairs line up. So you could have all of mom's homologous pairs lining up and all of dad's homologous pairs lining up, and cleanly there's all of moms, there's all of dads, moms, dads, right? Nice and simple. Again, this is not even taking into consideration crossing over or anything. This is just talking about where they're actually lined up. But here they could also line up opposite, where they could have mom, dad, mom, dad, right? And therefore now you have an even more shuffling of genes. Again, not even taking into consideration that crossing over will occur up here. So within that, you are now increasing the genetic variability even more so, because now you have mom, dad, mom, dad, mom, dad, mom, dad in each one. So let's just say this is the sperm that's chosen versus this one. This one would have a little bit more of both parents versus just one, where it would be more so another parent.